On the record. Okay, great. So yes, we're going to talk about post-COVID-19 syndrome and post-COVID-19 vaccine injuries or side effects, and we're going to see how we can manage them. And we know that both have common pathology. Both of them, they express the spike protein, and the spike protein blocks the ACE2 receptor, which is very critical because ACE2 enzyme is very important to convert angiotensin-2 to angiotensin-1,7, which is the anti-inflammatory arm of the renin-angiotensin system. And so we know for the fact those virus gain the function, or the gaining function research, which is those viruses are blocking critical enzyme on our body, which is very critical because blocking angiotensin anti-inflammatory arm of angiotensin system can lead to cytokine storm and can lead to pro-inflammatory cytokines that will lead to all these complications of COVID-19 infection. And also because of all this cytokine storm leads to disturbance in the mitochondrial function, endothelial function, and the autonomic nervous system. And that's why we have this post-COVID-19 syndrome. Vaccine does the same thing because they express the spike protein. The spike protein inhibits the ACE2, so they inhibit the anti-inflammatory arm of the renin-angiotensin system, leading to overactive of the inflammatory arm, which is mediated by angiotensin 2 type 1 receptor. And that leads to expression of interleukin-6 and polarization of the immune system into Th17, and then you would have what we call the neutrophilia, and you will have neutrophilia pneumonia, neutrophilic vasculitis all over the body, and that leads to all this organ damage and failure. It also stimulate those interleukin-6 can stimulate the platelets, so it leads to increase of D-dimers and clotting. And we know that recently that FDA did restrict Johnson & Johnson vaccines because they find out they express too much of spike protein that leads to clotting and coagulation and stroke. And that's why now it's officially that FDA restrict the use to certain people, but in the United States, but they are allowing it to be sold with no restrictions overseas. It seems like, you know, other people of other world or from other countries, they are not humans, and it's okay if we sell them Johnson & Johnson vaccine, but in the United States is being restricted. Are you adding? Because I see people online.
Yeah, I don't have anybody.
You got 35 people now. I told you it's going to be more than 30, right?
Yeah, I don't have any.
Because I invite my crowds. That's why.
Right. I don't have anybody in the waiting room that I can see.
I just add Dr. Klatt just now.
All right.
Okay, so let's go ahead and let me see here. So this presentation aimed to comprehensively describe the etiology, pathophysiology, clinical feature, diagnostic methods, and the latest novel therapeutics in management of post-COVID-19 infection and vaccination syndromes. A large proportion of patients discharged after being hospitalized for COVID infection or after receiving the vaccine experience. We know it's the same pathophysiology, which is the spike protein. With resistance COVID-19 related symptoms or complication, post-COVID-19 syndrome or post-COVID-19 vaccine injury involve varieties of immune alterations. It includes lymphopenia. Why? Because you would have that Th17 hyperimmune response and polarization to neutrophilia, and that will deplete the whole immune system. In a state of producing lymphocytes, they start producing neutrophils, and neutrophils cannot kill those cells which be infected by the virus. You really need to have cytotoxic T cells. So you need to polarize the immune system to Th1, cytotoxic T cells, M1. But it seems that when the macrophage eats those virus, the COVID-19, and because of the spike protein, those macrophages, they have receptors as well. You know, and they have ACE2 receptor, and same thing like any other cells. Those macrophages can respond to the spike protein by blocking the anti-inflammatory arm of the renin-angiotensin test system, leading to overactivation of angiotensin 2 type 1 signaling, and that leads to the polarization of the macrophages into M17 and then to Th17 and neutrophilia. And then you would have what we call the neutrophilic pneumonia. So it is very important to understand the immune modulation and the immune system and how we polarize the immune system, why we have neutrophilia, why we have eosinophilia, why we have basophilia and eosinophilia. So we need to explain the whole polarization of the immune system, including the adaptive immune system. And that starts from the macrophages. And the whole immune polarization and direction is based on what the macrophage eats. So if the macrophage eats bacteria, then what would happen? The macrophages breaks the bacteria proteins into peptides and protein fragments. And then those fragments will react with toll-like receptors on the macrophages. So the macrophages do have tongues, they taste the bacteria proteins. And when they taste it, they sit the bacteria proteins, after it's being degraded into peptides, throw toll-like receptors on their surface. So toll-like receptors, they are the tongues of the macrophages. That's how they sense that this is bacteria. And what would happen when those peptides and those fragments of protein of bacteria react with toll-like receptor on the macrophages? They polarize the macrophages into M7, and then M7 will start secreting interleukin-6, and then stimulate the polarization of the naive T cells into Th17, and then Th17 will start secreting interleukin-17, and interleukin-6 and interleukin-17 will polarize the white blood cells and make them or move them into producing eosinophil and to neutrophils. So the M17 leads to Th17, and then all the cytokines that's coming out from macrophages and Th17 leads to polarization of WBC into neutrophilia. So what we have when we have neutrophilia, when you do CBC differential and you see high neutrophils, then you would think the polarization of the immune system is to Th17, M17. Okay, and that's mean you may have a bacterial infection or fungus infection. Okay, so when the macrophages eats infected cells with viruses or cancer, what would happen? They break down the protein of viruses and cancers, and those protein turns into peptides and protein fragments, and then they react with toll receptors on the macrophages. And that signal the macrophages to polarize into M1, and then M1 starts secreting interleukin-2 and 12, and they polarize the naive T cells into Th1, and then the Th1 starts stimulating the activation of cytotoxic T cells. And so cytotoxic T cells is responsible to destroy those infected cells with viruses and those cancers by attacking them and releasing all those toxins and open up pores into their membranes called perforins and blow them up. So in order to kill virus-infected cells or infected cells with viruses or cancer, you need to have cytotoxic T cells. And that's why you will see lymphocytosis. Lymphocytosis is due to CD8 cells, right, are increased and they are battling and destroying either cancer cells or virus. So when you have lymphocytosis, then you think, oh, this could be cancer or viral infection. This is general rule. This is the holy grail of the immune polarization. Of course, there is exceptions, and we're going to talk about those exceptions in the coming slides, and possibly in cases like Dr. Patel. She has a case, and she told me that the case with lymphocytosis and basophilia or eosinophilia, and we're going to talk about it, which has to do with that the lymphocytosis here. It's not the CD8, it's the CD4, Th2. Now, what about if the macrophages eats the self-antigens, right? If they eat self-antigens and they break them down into peptides and proteins and fragments, they will react with toll-like receptors on the macrophage and polarize the macrophage into mRegs, and then mRegs will secrete the interleukin-10, and what would happen? They will polarize the naive T cells into Tregs, and then interleukin-10 will suppress all those effector cells, the Th1, Th2, and Th17. But what would happen if the macrophages eats the other genes or parasites proteins. Those proteins will be broken down into peptides and fragments. They react with 12 receptors on the macrophage and polarize the macrophages into M2, and then M2 starts secreting interleukin-4, and then that will lead to polarization of the naive T cells into Th2, and then Th2 will cause what? Differentiation of the CBC into eosinophil and basophil. So this is the holy grail. This is the basics. This is the language that we need to learn in order to really understand what's going on with COVID-19, because COVID-19 disease, it is Th17 immune dominant disease. And normally, when the macrophage eats infected cells with viruses like flu viruses, polarization is supposed to be lymphocytosis, cytotoxic T cells, and they were going to kill those infected cells with viruses. But what happened is this COVID-19 hijacked the renin-angiotensin system, and on those macrophages, you will see ACE2 receptor, and they block them, leading to angiotensin 1, angiotensin 2 type, angiotensin 2 type 1 receptor signaling to be overactive, which leads to expression of interleukin 6, and that leads to polarization of the immune system to Th17 and neutrophilia. And neutrophils cannot do much into destroying those infected cells with viruses. In fact, this overstimulation of neutrophils leads to neutrophilic pneumonia and destroy your own self, blood vessels and lung, and leads to all this pneumonia we call neutrophilic pneumonia. And also it leads to neutrophilic vasculitis and stimulates the coagulations, the platelets activations. It's all mediated by interleukin 6 that's being overproduced because of the wrong or polarization of the immune system to wrong direction, which is toward Th17. Th17, we use it for killing bacteria and fungus, but not for the virus. And this is how your immune system is being fooled by the virus. And of course, this is engineered. They know this is going to happen. And it's sort of immune suppression because you are pushing and polarizing immune system in the wrong direction by hijacking the renin-angiotensin system, which leads to polarization of immune system Th17 and eventually neutrophilia. So that's the COVID-19. So knowing that COVID-19, it's Th17 dominant immune disease, then we can manage the COVID-19 based on that. Then we need to find the drugs that inhibit the Th17 and inhibit the interleukin 6 and inhibit interleukin 17, whether it's going to be the FDA approved antibodies, we have many of them. They can inhibit interleukin 6 receptors or interleukin 17 receptors, or we use repurposing drugs like ivermectin, hydroxychloroquine, methylene blue, serotonin reuptake inhibitors. All of this can help the expression of interleukin 6. So the mechanism of action of these drugs is inhibiting the interleukin 6 expression, is inhibiting the polarization of the immune system to Th17. So let's go ahead and look at Dr. Peter. Let's see here, Dr. Peter Hortis. He's the
We also took on a decade ago the interesting problem of making
left-wing doctor that Dr. Fauci and all other doctors, they based their information on what Dr. Peter Hortis is saying. And I admire him in this presentation in the beginning of COVID-19. He was very honest, very sincere in everything he said. And we're going to learn what he's saying, and I confirm everything what he's saying. But then after he got the funds and he started developing vaccines, there's a lot of disinformation, and he is calling us that we are doing the disinformation, but we're going to go through his videos and find out who is doing the disinformation. So anyway, let's start with Dr. Peter Hortis' presentation here at time at the beginning of the COVID-19 pandemic, and how he is explaining to the audience here that there is difficulty in developing those vaccines because there's a problem with safety. So that's a good thing. And there's many things that we're going to learn from him. And I really admire everything.
Coronavirus vaccines, because we recognize these as enormous public health threats, and yet we have not seen the big pharma guys and the biotechs rushing into this space. So we we partnered with a group at the New York Blood Center and the Galveston National Laboratory to take on the big scientific challenge of coronavirus vaccines. And I say a scientific challenge because one of the things that we're not hearing a lot about is the unique potential safety problem of coronavirus vaccines. This was first found in the early 1960s with the respiratory syncytial virus vaccines, and it was done here in Washington with the NIH and Children's National Medical Center, that some of those kids who got the vaccine actually did worse. And I believe there were 2 deaths in the consequence of that study, because what happens with certain types of respiratory virus vaccines, you get immunized, and then when you get actually exposed to the virus, you get this kind of paradoxical immune enhancement phenomenon. And we don't entirely understand the basis of it.
So this is very important part of our past experience in developing vaccine against respiratory syncytial virus. And it's similar to COVID-19, that this vaccine, the proteins that they express, they polarize the immune system into Th17 and into possibly Th2. So away from cytotoxic T cells, the same like COVID-19. So it's fooling the immune system. And what would happen when you have that? Then you would have this hyperimmune response, which is Th17 hyperimmune response. And also, because of polarization to Th2, you would have a lot of antibodies. But those antibodies can neutralize those 3 viruses, but they cannot do much in neutralizing and killing and destroying and clearing those cells infected with viruses. You need cytotoxic T cells. So because of this polarizations into either Th2 or Th17, which will lead either to eosinophilic pneumonia or neutrophilic pneumonia, depending on which one is dominant. And that's the problem with syncytial virus vaccines. Of course, he cannot explain it because I don't think he has a clue about that we have 2 kinds of inflammations, Th1, Th2, Th17, and I don't think he understands that those syncytial virus proteins, when the macrophage eats them, those fragments react with toll-like receptors on the macrophages and signaling the polarizations into Th17 and Th2, leading to eosinophilic pneumonia or basophilic pneumonia. We're going to see that in the coming slides. When he's trying to perfect the COVID-19, he talked about eosinophilic pneumonia, but we're going to talk about it in the coming slides. So that's the problem. Okay, so that's the problem of failing of this vaccine. Similar what's going on with COVID-19, because those spike receptor, they do the same thing. They polarize the immune system to Th17 and leads to a neutrophilic pneumonia. And it's only 2 deaths, and 2 deaths, not 14,000 deaths. That's happening with COVID-19, 14,000 deaths, which is 0.002% of people received the COVID vaccine died from the vaccine. And he denied that. And when we go in the coming slides, he denied the death of any death from the vaccine, but it's being reported in CDC. It's 0.002 of the death from the vaccine. Now, the death from COVID virus itself is 0.2. And he can argue, yes, the death from COVID virus is 100 times more than death from COVID vaccine, and that's true. But then, and then that's why he say, well, it's better if we take the vaccine versus, facing the virus itself. And that's true in his perspective. But if you go to perspective of Dr. Peter McCullough, he will say, well, I can reduce the death 100 times if we start early treatments. So I will even produce the same thing like what the vaccine is doing with almost no much of that money, like trillions of dollars in developing vaccines. Of course, that's not a good news for the big pharma, for some doctors to say early treatment will reduce the death of the COVID-19 100 times, which is equivalent to the vaccine. And this will bust the trillions of dollars that the big pharma is going to produce. And that's why they hate Dr. Peter McCullough, because he's not preaching what's line up with the big pharma. And I think you'll be pleased more if you are helping the interest of or you're preaching and you are presenting things that lines up with interest of the big pharma. Anyway, let's go ahead and finish up what he's saying. But there's only 2 deaths, and they stopped this syncytial respiratory syncytial virus.
But we recognize that it's a real problem for certain respiratory virus vaccines. That killed the RSV program for decades. Now the Gates Foundation is taking it up again. But when we started developing coronavirus vaccines and our colleagues, we noticed in laboratory animals that they started to show some of the same immune pathology that resembled what had happened 50 years earlier. So we said, oh my God, this is going to be problematic. But we collaborated with a unique group that figured out how to solve the problem. That if you narrow it down to the smallest subunit, the piece that of what's called the receptor binding domain that docks with the receptor, you get protection and you don't get that immune enhancement phenomena. So we were really excited.
So let's talk about what he did. Okay, so they took the spike protein and they clip it. What they clip, they remove all the epitopes that will lead to polarization to Th2. And they left only the piece of the spike protein that reacts with the receptor, which is the ACE2. But the problem is, if you block the ACE2, you interfere with the renin-angiotensin system, and you lead to overactivation of angiotensin 2 type 1 receptor signaling, which leads to expression of interleukin-6 and polarization to Th17. And interleukin-6 itself is a pathophysiology that is behind the neutrophilic pneumonia. So with his method, he may be able to clip all those epitopes that leads to hyperimmune response to Th2 and eosinophilic pneumonia. That's true. That's what he did. He solved that problem of polarization to Th2 by clipping all those epitopes that leads to stimulation of the macrophages into M2 and Th2, but he did not solve the problem of blocking the ACE2, which leads to polarization of the immune system to Th17. So he still have the problem. And maybe he solved the problem of eosinophilic pneumonia, but he did not solve the problem of neutrophilic pneumonia and neutrophilic vasculitis. And that's the problem with the vaccine development. The other problem is that you are polarizing the immune system to Th2 and Th17, and you're producing antibodies. And that's antibodies that they can neutralize the viruses. And they base the success of their viruses based on the antibody titers, okay? But that's not the way we determine the success of the vaccine, because it's not just enough to have enough antibodies or high antibodies. Even high antibodies and polarization to Th2, it's not good, because that's mean you have too much of interleukin-4, which will suppress your cytotoxic T cells and Th1. So you need to have a vaccine that gives you a balance. It gives you some antibodies, but a lot of cytotoxic T cells. So really the vaccine that's effective is the vaccine that can give you Th1 and cytotoxic T cells. And that's not being measured. They don't care about it. They just prove that to the public or to the FDA, hey, we have a lot of antibodies here, see the vaccine. But that's not the way that we measure the efficacy of the vaccine. You really need to have both the Th1 immune response and Th2 antibodies and cytotoxic T cells. And if you have too much of antibodies, that's a bad vaccine, because you're suppressing the cytotoxic T cells. Now, what will happen? Well, first of all, those vaccines will not stop the transmissions. And the reason, because those vaccines be introduced intramuscular. So you're getting all this IgG, but you're not getting any IgA. So it's better if those vaccines be introduced intranasal to the mucosa, so that you can get IgA. So that's one. So you're not getting, the vaccine will not stop the transmission. Although in one of his lecture, he's saying those vaccines is stopping the transmissions, and that's disinformation. Now, the other part is that, those vaccines is giving you antibodies, and it will neutralize the free viruses, but will not destroy those infected cells with viruses. So what would happen to those viruses? You are putting pressure on them, where they start mutating inside those cells being infected and change into variants. So the vaccines actually leads to pressure evolution of development of more variants. And that will prevent us from getting into the herd immunity. So that's the problem with the vaccine. The whole variance is being pushed by the vaccine, because the vaccine is not clearing the cells from the virus. And we're going to see that and learn that from the coming slide, where Dr. Soon Patrick, he's from South Africa, he's a billionaire, the most intelligent doctor that I know. He's like Elon Musk. I think he's an INTP. And he's explaining that we need a vaccine that gives you Th1 and cytotoxic T cells to clear those infected cells with virus and prevent the pressure evolution of the variants and preventing us from getting into the herd immunity and prolonging the whole epidemic to 2 years. I think if we develop the right vaccine that gives us Th1, and we develop a vaccine that does not block the ACE2 receptor, maybe we use the capsid of the virus instead of the spike protein itself, we may be having better vaccine, but we don't have that really good vaccine. The vaccine that developed is all based on the spike protein, and the spike protein is pathologic. You cannot really develop vaccine from toxic or toxins. And very important that people need to know here that those spike protein, how they came up from, I mean, where they come from. The idea of developing spike protein, it came from the snake venom. So snake venom is the way that we can get peptides, antihypertensive drugs, captopril, enalapril, lisinopril. So lisinopril, captopril, enalapril are peptides originated from the snake venom poison. So we are developing medication from the snake venom. And they block the ACE receptor, not ACE2, but ACE receptor. And ACE receptor, when you block it, prevent the conversion of angiotensin 1 into angiotensin 2, which will prevent, producing this angiotensin 2, which causes hypertension, right? So it's preventing that, so that lower the blood pressure, which is good, so that we use for medicine. But in developing the COVID spike protein, same thing they developed from the vaccine, but they block the ACE2, which is the enzyme that convert angiotensin 2 to angiotensin 1,7, and angiotensin 1,7 is responsible for anti-inflammatory and vasodilation. It's the opposite of angiotensin 2. So if you block that, you're blocking the vasodilation and anti-inflammatory, allowing this inflammatory signaling mediated by angiotensin 2 and angiotensin 2 type 1 receptor to lead to all these problems, which is the cytokine storm and interleukin-6 overproduction and polarization to TS17. So that's how they engineered, or they came up from the spike. It's come from the poison, because there's no much difference between ACE enzyme and ACE2. It's almost similar. And it's so easy to tweak those peptides or create peptides from the venom that will block the ACE2. The same way we develop peptides that blocks the ACE and we use for medicine, we can also develop peptides and proteins or spike proteins from the snake venom to block the ACE2 and poison the whole universe. And that's the gain of function. It's a biological weapon. So let's go ahead and finish up what Dr. Peter Hodes is saying here about that we need enough time to pass the safety. This is before he received the fund, okay?
About that, and we proposed this to the National Institute of Allergy and Infectious Diseases. They funded it, and we wound up actually making and manufacturing, in collaboration with Walter Reed Army Institute of Research, a first-generation SARS vaccine. So SARS was the one that emerged in 2003. And then this new one, of course, we call the SARS-2 coronavirus. We had it manufactured, but then we could never get the investment to take it beyond that. So that was really unfortunate, because we had the vaccine ready to go, but we couldn't move it into the clinic because of lack of funding, because by then nobody was interested in coronavirus vaccines. When the Chinese started putting up the data on bioRxiv in January, February, we saw very close homology between the two and realized that we may be sitting on a very attractive coronavirus vaccines. Now we're working with NIH, and we'll work with BARDA and others to get the funding, but now we'll have that lag. And these clinical trials are not going to go quickly because of that immune enhancement. It's going to take time. So, and unfortunately, some of my colleagues in the biotech industry industry are making these inflated claims. You know, you've seen this on the newspapers. We're going to have this vaccine in weeks, or in this and that. What they're really saying is they can move a vaccine into clinical trials, but this will not go quickly, because as we start vaccinating human volunteers, especially in areas where we have community transmission, we're going to have to proceed very slowly, very cautiously. The FDA is on top of that. They have a great team in place at the Center for Biologics Evaluation Research. They're aware of the problem, but it's not going to go quickly. We're going to have to follow this very slowly, cautiously, to make certain we're not seeing that immune enhancement. So, you know, now we're hearing projections a year, 18 months. Who knows? This is not going to go quickly. The bottom line is, had we had
that's the minimum, right? The reality is that Trump did push the whole thing into less than 1 year, less than that, maybe a few months. And they say, okay, we go emergency permission license, and they got this emergency license, and they start putting it on people, and we got almost 0.002% death, which is 14,000 among 500 million people being vaccinated here in the United States. So that's the data. Okay, so that's the problem. So the safety was not being, looked at, and they pushed the whole thing, and we end up with this death. And also, not just that, the number is going to be very high with the side effects that and triggering subclinical chronic disease to become chronic diseases, or even diseases out of the blue. So maybe somebody has subclinical multiple sclerosis, but because of the vaccine, they have full-blown multiple sclerosis. Maybe this multiple sclerosis, which is preclinical, it's supposed to manifest at the age of 80, it manifests at the age of 50. And it's because of the vaccine. Still, the vaccine is blamed, even if they have subclinical and genetic risk of having chronic disease. So this is the problem that we have with the vaccine. It's because of sticking into developing vaccine based on the spike. And I think that's the mistake here. We may be able to create vaccines from other parts of the virus more safe than the spike. And we're going to learn that from the coming slide from Dr. Patrick Soon, which is a South African doctor. I like this doctor. But let's go ahead and finish what
investments early on to carry this all the way through clinical trials. Years ago, we could have had a vaccine ready to go.
Okay, so let's go ahead and check what Dr. Patrick Soon is talking about, developing a vaccine that leads to memories of Th1 and cytotoxic T cells, not just Th2, because Th2 vaccines that leads to antibody-based is not enough, and it may actually cause problems, and it can cause pressure evolution of developing variants. So we're going to learn that from Dr. Patrick Soon and how he developed a vaccine, not based on the spike, but based on the other parts of the virus, the capsid. And I think Dr. Patrick Soon did nail the whole idea of developing a Th1-based vaccine. Let's hear what he's saying.
What do you think of boosters? Will this increase efficacy against, for example, the Delta variant?
Well, that's what we're actually pursuing. And the good news is we're in a phase 1, 2, 3 trial right now. And we've decided to go into the heart of the storm where, you know, obviously the initial variant, now the Delta variant, and soon the Lambda variant. And this inevitability of the viruses changing against antibodies means that you need both an antibody and a T cell. So I'm excited to tell you that we've launched a phase 1, 2, 3 trial in South Africa against and boosting both the Johnson & Johnson and the Pfizer vaccine. And the first patients will probably be injected or jabbed this week.
Now, I felt pretty good with the fact that I got double vaccinated by the time the summer started, but of course, Delta is a different challenge. So when we think about the booster shots, I had a sobering conversation on another show yesterday with someone from the space, and basically said they're not sure that a third mRNA shot is the ticket here, that it's going to have to be something else in the therapeutic space, a different way of going at it. Is this sort of the point that we're entering now in the pandemic?
Well, I've been, screaming at the top of my lungs for the last year that just trying to block the virus with an antibody is insufficient. You have to actually kill or clear the infected cell, not only to prevent transmission, but to actually give you long-term immunity. And the innards of the virus, called the nucleic capsid, is what we've actually done. And I'm excited to say that our preclinical data show that, in fact, we protect all the way to the Delta variant. So I think the combination of an antibody vaccine and a T-cell vaccine is going to be the answer.
Why doesn't the FDA fully approve the COVID-19 vaccines out there? We've got so many people who are hesitant to take the vaccine. A lot of them surely are concerned about, because of misinformation that they've seen on social media, but others may be looking at the FDA and saying, I'm not going to take it until they approve it.
Well, I think the issue with regard to vaccinology is really long-term immunity. So, I can't speak to what the FDA's decision-making process is, but I do believe, first, that everybody should get the vaccine because it definitely reduces death risk. But we need to go to what I call now the second-generation vaccines. And as we said, Immunity Bio is now taking the approach that we will become the universal boost. I think long-term, we need to figure out an RNA and a DNA vaccine combination where the RNA looks as if it gives you the best antibodies and the DNA looks as if it gives you the best T cells. So if one can then create what we call a heterologous vaccine. But right now what we're doing, we're taking our DNA vaccine and at Immunity Bio and making that the third booster, so to speak, of both the Johnson & Johnson and Pfizer vaccine in South Africa.
To get people on board with this, is there a mind shift that's needed in the sense that for a while we were hopeful that this first round of vaccines, whether it was from Pfizer or Moderna, were the answer? Did they simply buy us time to get to other solutions, like the kind of things you're working on?
Well, that's exactly what I was concerned about, right? I mean, this first vaccine, the first generation of antibody-based vaccines, is really important because it actually would stop the death rate, reduce the load on the hospitals, but it doesn't really affect transmission. Meaning the ongoing pandemic. And what's worse, the transmission, and those are unvaccinated, as you could see from the results, will continue this pandemic. And then if you add to that concept that the virus will just mutate against an antibody. So it's so important and so urgent that these next second generation vaccines now come to fore. Unfortunately, with the supply chain being sucked up by the first generation vaccine, we're in a sort of vicious circle where we can't produce enough at the second generation in order to enable this to come to the fore. And that's really where we are actually at Immunity Bio. So we've taken the position that we need to go country by country at a time. So we started in South Africa.
I'm sure it's way down on your list of concerns, Doctor, but you famously own a stake in the Lakers. And I just wonder what you think about fans being able to come to the games this year. You've just added Russell Westbrook in the offseason. He's surely going to be a draw, but are people going to have to, wear a mask while they watch?
Well, even more so. You see, this is the other disconcerting fact. When you look at it both scientifically and logically, when we were concerned very early on, the very early stage of the pandemic, of what you call asymptomatic infections, that you would be asymptomatic and could spread, then all of a sudden, CDC said, wear masks. Guess what? When we get the vaccine, or people get vaccinated versus those that are unvaccinated, and the person that's vaccinated, that's infected, this Delta variant actually has 5 to maybe 1,000 times more viral load in your nose. So it'll infect the unvaccinated. And even those who are fully vaccinated, as you know, it's people that have what we call breakthrough infections. So the concept of now not wearing masks is really a little illogical. It's actually worse now than it was even at the beginning of the pandemic. So the answer
unless,
is
Doctor, unless you say, players have to be vaccinated and you've got to be vaccinated to get tickets to come to the game.
Vaccinated doesn't mean you're not going to be, you don't prevent transmission. That's the whole point.
But you're not going to end up in the hospital. You're not going to take a bed away from a cancer patient. Yeah,
but putting a mask on is not just for yourself; it's for your fellow man. So putting a mask on is prevent transmission to others, so we can stop the cycle of pandemic, right, of infection. So no, look, I personally advocate masking when you're inside or outside. It may not be the.
I mean, we're not saying everything is saying I like everything, but there's some points where he said that so we know that the vaccine does not prevent transmission, and that's true, and he confirmed that, which is good. So that's one point. Now, lowering the risk of dying from the COVID-19 virus versus the vaccine is 100 times, and that's what he's trying to present. That taking vaccine is you die the 100 times less than facing the virus itself. But then the other side, which is the red side of the aisle, Dr. McCullough will say, well, I can reduce the death rate from facing the virus to 99% to 1% death rate, which is equivalent to what the vaccine can do just by early treatment by all these repurposing drugs. So he has a point there, and it's more efficient. And then this natural infection, you would have mucosal immunity, so you can prevent transmission if you are recovered from COVID virus infection, because you do have immunoglobulin A in your mucosa. So there is no much, if you're covered from COVID-19, you're not transmitting the virus as he indicated. Versus the vaccine is not giving that protection against transmission. And the other part of the vaccine is pressuring the virus to evolve into variants. And that's a problem where he's going to solve it with his TH1 vaccine. But Dr. McCullough will say, I can early treat those patients and they get their natural immunity. They prevent the transmission and we can reach the herd immunity. And we're not getting this 100 times more than the vaccine death rate. We can do that with early treatment, and that will reduce the death rate versus the vaccine. So that's the argument of both aisle. I think your osteopathic doctors you will agree more about early treatment. It's more effective, more efficient. You get the herd immunity, and you can get the same level of the vaccine. But the people who are developing vaccine, big pharma have interest there. Definitely, they don't want early treatment because that will destroy the purpose of developing a vaccine, which is lowering the severity of disease. And if that early treatment accomplished that, then there's no much purpose there, because even transmission, it's better to be recovering from the natural infection versus the vaccine. So that's the argument of both sides, and we're just learning it from
advocated
now
in our country.
Let's learn about Dr. Peter Hotez when he is developing the vaccine, and he trimmed the spike protein. As I mentioned, the spike proteins came from the snake venom. That's the gain of function that blocks the ACE2. But also those spike proteins that in the virus or that being expressed by the vaccine, they do have epitopes that may lead to polarization of the immune system to Th2. So he said, okay, we're going to take this and we're going to trim them. The one that produce epitopes that polarize the macrophages into M2 and Th2 and give you eosinophilic pneumonia. But he forgot that blocking the ACE2 will lead to polarization to Th17, lead to neutrophilic pneumonia. So let's go ahead and see that he is so excited and happy that he solved the problem of eosinophilic pneumonia and hyperimmune response from Th2. But the problem is that he did not solve the problem of neutrophilic pneumonia mediated by Th17 hyperimmune response due to blockage of the ACE2 enzyme. So let's go ahead and learn that exhibit.
Okay, thank you. Well, thanks for having me, and sorry we had a couple of technical difficulties, but
just 2 minutes, okay?
Permission to move forward in the lab. So the great thing about these receptor binding domain vaccines, the spike protein, they seem to induce high titers of neutralizing antibody depending on the adjuvant that you use. It seems to remove a lot of the immune enhancing spike protein epitopes, and as I said, this yeast expression system. And when we initially looked at this, it was amazing how quickly the scientific community was responding, putting out
their data on bioRxiv. I've never seen anything quite like this, but we were able to ascertain pretty quickly that SARS-1 and SARS-2 look pretty close. So we thought we're going to try as best we can to do this in a staggered way. We have the receptor RBD1 ready to go now, and then we'll advance the RBD2. And this is our RB1 receptor binding domain SARS-1 candidate. We call it, it's got the very memorable name of RBD219N1. And the reason we call it N1 is our scientists found that if they express it without the N-terminal amino acid, then we get higher levels of expression yields. And also, one of the problems with Pichia pastoris, one of our lead workhorse expression vectors, is they can over-glycosylate. And by getting rid of that N-terminal amino acid, we did not affect the conformation of the protein, but got rid of a lot of the glycosylation window dressing, and we were getting really good expression yields. So we were able to show in a transgenic mouse model, working with the Galveston National Lab, they have a mouse model with expressing the ACE2 receptor, the angiotensin-converting enzyme 2 receptor. They also got a mouse-adapted SARS-1 coronavirus. We were able to induce very high levels of neutralizing antibody. And interestingly, we found an alhydrogel, straightforward alum, was one of the best adjuvants in inducing protective immunity. This is the survival curves of our RBD219N1 construct, our complete survival versus total death among our mice that were vaccinated with the adjuvant alone. And this is, people talk a lot about immune enhancement, but not many people actually show what it really means. This is what it looks like. This is using looking at histopathology in the lungs of our vaccinated versus control mice. And those brown dots are immune histochemistry of eosinophils. And you see that with the full spike protein kind of reproducing Lenin's original observation, lots of eosinophilic infiltration. But if we can get the alhydrogel right, we can completely prevent eosinophilic immune enhancement. So we were pretty excited about this because we had a vaccine that worked and it was safe. And now Lanying has taken...
The vaccine is safe, that's not true. And yes, he solved the problem of hyperpolarization of immune system to Th2 and eosinophilic pneumonia, but he did not solve the problem of hyperimmune response of Th17 and interleukin-6 and neutrophilic pneumonia. So that's the problem with the vaccine. So adding adjuvant like interferon gamma, thymosin alpha to the vaccine, possibly that may help to polarize the immune system into Th1, because they did it for influenza virus with success. So I don't know why they're not adding that, because that may help to push the macrophages from polarizing into Th17 into macrophages from M17 to M1 and then Th1 and cytotoxic T cells. So this may be, we may add that interferon alpha, gamma, thymosin alpha may lead to polarization of Th1, and that will prevent the polarization to Th17 and neutrophilic pneumonia. But nobody, I don't know if they thought about it, but he doesn't have a clue about Th17. He's those old school people, and I was one of them before we learn about Th17. We only know Th1 and Th2. So I think he's still in that circle, and he did not break through. Understanding that there's also Th17, which is mediated by the ACE2 blockage, which is blocking the anti-inflammatory arm of the angiotensin-2, allowing renin-angiotensin system, allowing the angiotensin-2 to overactivate the angiotensin-2 type 1 receptor signaling that leads to interleukin-6 expression and neutrophilia and hypercoagulation, and then neutrophilic pneumonia and neutrophilic vasculitis, which leads to this multi-organ failure that we see in patients, whether they are receiving COVID vaccine or they are facing the virus itself. Okay. So let's see about this information here, and where the media, the CNN and the left-wing docs and the media and BBC, whatever, from where they learn that ivermectin and hydroxychloroquine are drugs for treating malaria and parasites. Okay, and they're using ivermectin to treat horse parasites. From where this idea came from? Yes, it is. That's true. You know, ivermectin and hydroxychloroquine does have antibiotic effect, and they kill the malaria, and we use it for treating parasite, the worms, and so it has anti-parasitic effect, and also it has anti-malaria effect. We generally would say antibiotic, but also what he's missing, that those two drugs, they also have anti-inflammatory effect. That's why we use FDA approved ivermectin to treat rosacea, because rosacea is a skin disease mediated by Th17 and neutrophilia. And we give ivermectin. It's the only effective drug that suppress the Th17 interleukin-6 expression is using ivermectin topical. It's FDA approved. And my wife is using it. And I think my wife is a human, not a horse. And hydroxychloroquine is being used as an anti-inflammatory to treat systemic lupus erythematosus. It's an FDA approved because SLE, it's Th17 dominant immune disease. But because of the ignorance and disinformation of those left doctors who have no clue that those drugs has anti-inflammatory effect, especially Th17 anti-inflammatory effect. We know that our group, I think everybody here, we learned that there's three kinds of inflammation. There's Th1, Th17, Th2. They're not just one inflammation. And in order to manage disease more effectively, especially inflammatory disease, autoimmune disease, we need to learn which inflammation they have. Is it Th2? Is it Th1? Is it Th17? And maybe you have mixed. You may have Th1 and Th17, right? Like multiple sclerosis. Or you may have Th2 and Th17, like celiac disease, for example, or psoriasis or rosacea. You have a lot of Th17 there. So we can give drugs that inhibit the Th17 without affecting Th1, that's very important, because if you give steroids suppress everything, including Th1, and that put the patients at risk of having cancer and viral infection. So you don't want that. You just want to block that inflammation that's being active, which is Th17. And let's go ahead and see the disinformation, a lack of knowledge with sarcastic, that hydroxychloroquine and malaria, it's only antibiotics, and it's only for parasites and malaria and worms, and it's not for other inflammatory disease, because he has no clue about what he's saying. And this will be in the section, I think.
An important story we're working on today that
833.
Because of the adverse effects with the vaccine, not because kids are catching COVID.
No, pediatric hospitalizations are way up. A lot of it is due to COVID. Also, we have a second virus pathogen circulating known as respiratory syncytial virus, or RSV, and we're even seeing kids now with combined RSV and COVID, and masks can help with both of those illnesses. But no question, we're seeing a big rise now in pediatric hospitalizations from COVID. None of it is due to the vaccine.
More people have died from the COVID vaccine than COVID-19.
620,000 Americans have died of COVID-19. To my knowledge, no one has died from a COVID vaccine.
And no knowledge.
If you take hydroxychloroquine, ivermectin, and zinc, you can beat COVID easily.
No, if you take hydroxychloroquine and ivermectin, you will have some protection against malaria and against parasitic worm infections, but it won't do anything for your COVID.
See that?
Your immune system is 99.9% able to beat COVID, and it's the vaccine that actually lowers your immunity.
No, that's not true at all, and that's why 620,000 Americans have lost their lives, and because the immune system could not handle the virus. And the fact that we are seeing this dramatic increase in hospitalizations from unvaccinated people.
And that's because Dr. Peterbakal will say all this death is because of lack of protocols of early treatment. And at the beginning, those patients who have COVID, they tell, stay at home until you get respiratory failure and your oxygen is low, and then they end up with hypoxic pneumonia. And that's the problem, lack of early treatment. If we have the early treatment protocol, we can reduce the death rate the same and equivalent to what the vaccine can do. But Dr. Peter Hotez does not recognize early treatment, and because early treatment will negate the interest of doing the vaccine. Okay, so is it too much? Is it too heavy? Or you want me to continue, Dr. William? Are you there? Can you hear me, guys?
Keep going.
Keep going? Or
Keep
you want to
going.
have some break and some QA? And
There
do
is-
other sessions? You want to do other sessions?
Well, we'll do other sessions, but we keep going for now.
Okay.
There was a question here. What was the third type of inflammation?
So we have three kinds of inflammation. Th1 mediated inflammation like contact dermatitis, TB, those are Th1 mediated. And then Th2 mediated inflammation, where you have eosinophilia and basophilia, that's allergies, parasitic infection leads to Th2 inflammation, celiac disease leads to Th2 inflammation. And then the Th17 mediated inflammation is like psoriasis, rosacea, COVID-19. Those are Th17 mediated. Now there is some mix, COVID-19, there's some Th2 also immune inflammation, mediated inflammation, but it's more Th17 in case of COVID-19, because we know that ACE2 is responsible to break the bradykinins, and because it's being activated by the virus, so you have active bradykinins, which leads to
heat too.
Release of interleukin-4 and histamines. So you have some histamine syndrome in those patients. But it's more dominant of Th17 because we see neutrophilia, you don't see eosinophilia and basophilia. Maybe the original vaccine and that developed leads to more of Th2. That's why you see that in the case that the original vaccine was giving them eosinophilic pneumonia. But once they trim that, it's actually giving us a neutrophilic pneumonia. So that indicates it's more of Th17 than Th2, but there's some components of Th2 there. So most of the time, the autoimmune diseases and degenerative diseases and inflammatory diseases, you can see a mixed bag of Th1, Th17, like multiple sclerosis, or in ulcerative colitis, you will see Th17, Th2. Which one is dominant? You go to CBC differential, and if you find neutrophilia is dominant, that's mean it's more Th17. If you see that it's eosinophil and basophil, that's mean it's Th2. But when you see lymphocytosis, that's mean it's more of Th1. I mean, there is some exceptions there. Like Dr. Patel, when she brought the patient, 1-year-old patient with high CBC. Well, it's not that high. It's 18,000 for the kids at that age. The max norm is 17,000. But the patient is manifesting eosinophilia and lymphocytosis, and possibly the lymphocytosis is due to CD4, which is the Th2 overactivities, and that's why we need to measure the CD4-CD8 ratio to know exactly what kind of lymphocytes is causing lymphocytosis. So if it's CD8, that's meaning maybe they have some infection, viral infection that leads to lymphocytosis. But very clear that celiac disease is Th2 dominant and some of Th17 as well, but because of too much of eosinophilia there, so we can know that's going to Th2. So those are things we need to learn. And we're going to have maybe a complete day of presentation of just focusing on all these varieties of disease and their whether they are Th1 or Th2 or Th17 or both mixed. We need to really, any disease up there, whether it is degenerative, metabolic, we need to look and screen the metabolic or the immune polarization. It's very important so that we can give the right peptides and the right drugs to balance the immune system. In case of cancer, we all the time want to polarize the immune system into Th1, M1, and cytotoxic T cells, because that's the only way we can kill cancers. And the cancer are secreting chemicals to polarize the immune system away from cytotoxic T cells to Th17 and Th1. So we're giving drugs that polarize the immune system Th1N1 and blocks the Th17 and Th2, including methylene blue. You can use ivermectin, or you can use antihistamine in those cancer, or we use antibody blockades as well, PDL1 and CTLA4. That's complete another lecture. But it's very important to screen your patients with chronic disease, screen their immune polarizations, and knowing exactly what direction they are and try to balance it. And we're going to learn that through the sessions that we have here with Dr. William, or through my sessions in chronic disease prevention. And then also in chronic disease, you need to manage the metabolics, all the metabolic, including sex hormone, very important, especially post-COVID-19, with all the cytokine and interleukin-6 and neutrophilia. And this leads to mitochondrial dysfunction, endothelial dysfunction, and autonomic nervous system dysfunction. And that leads to the post-COVID-19 syndrome. So you need to really restore it. And in addition to this, the inflammation of COVID-19 and interleukin-6 and all those free radicals, it can damage the testis and the ovary, and can damage the brain, the pituitary glands, and it can lead to hypogonadism. And we saw cases of patients where they have low testosterone due to the vaccine or due to the COVID virus. So it's very important to check all your patients with post-COVID-19 syndrome or post-COVID-19 vaccine injuries with their sex hormones. It's very critical. I think Dr. Patel, she has a very interesting case about that. I think she presented, and the patient was having was a male with male menopause due to post-COVID-19 vaccine injury. Any other questions? Are we good?
Let's see. Vaccines have shown not to prevent the spread. No evidence that the vaccine has reduced death. In fact, now that's
well,
Dr. Khan.
Well, we need to make sure that we go with data. And the CDC said we have 0.2% died from COVID, and 0.002% died from the vaccine. And that's too much. It's 14,000. It's a lot. I mean, they stopped the vaccine program of the respiratory syncytial virus just for 2 cases, not 14,000. But that's how they minimize it. They say, just 0.002, and it's 100 times less death if you receive if you face the COVID. And the argument, this is how you argue with them. You don't argue with them with the data that they believe in. You tell them, hey, but if we do early treatments, we can reduce the death into less than 99%, which is the same and equivalent of what the vaccine can do. We can go from 0.2 to 0.002 if you start with early treatment, and that's proven fact. If you start methylene blue and ivermectin, which is, again, we're not using ivermectin because of its antibiotic, although you can argue that the ivermectin does block the spike receptor, okay, and preventing it from internalization. So that's antibiotic. And methylene blue does the same thing, and hydroxychloroquine does the same thing. But you can argue that we're using it also for its anti-inflammatory effect by inhibition of Th17 and interleukin-6 expression and polarization. So that's why we're using it. And of course, in COVID-19, it's good to use thymosin alpha, interferon gamma, anything that polarizes the Th2, Th1 and balance the immune system. It's great. And we have a complete ingredients, whether it's supplements or repurposing drugs, that we developed for post-COVID-19 and for COVID patients. So I think that's a better argument with the mainstream medicine doctor who will bring the facts from their own CDC. I think this is how you answer that in a way that we are, at least we have some common ground to discuss. Any other questions?
You were a big advocate of phototherapy also, at least early on. Where are you with that?
Well, that's what we started with. We know that the virus concentrate mainly in the salivary glands in the beginning, and in the esophagus. But salivary glands is really the place where they replicate and they proliferate. And so with methylene blue mouthwash, or even you can swallow the methylene blue because it does have antioxidant effect and it blocks interleukin-6. and you do light therapy, you can really enhance the antibiotic effect of the Methylene Blue. In addition, you get the benefit of its anti-inflammatory effect by blocking the interleukin-6 expression polarization to Th17, and it's very important. And plus, the Methylene Blue does help to restore the mitochondrial function, and that's part of the thing that we knew, especially with cytokine storm, with all these free radicals. It can definitely oxidize the cytochrome ions in the mitochondria and cause mitochondrial dysfunction. So you can restore the mitochondrial function using Methylene Blue. And Methylene Blue also helps to kind of involve in electron transport and bypass the blockage caused by the free radicals damage of the mitochondria. So it will restore the electron transport chain and the energy flow and production of ATP. In addition to that, Methylene Blue increases acetylcysteine, which is very important. Why? Because there's some theory that the COVID-19 blocks the nicotine receptors and cause autonomic dysfunction. But Methylene Blue blocks acetylcholinesterase, and that helps to prevent the blockage of the nicotine receptors. And in addition to that, Methylene Blue, it's a serotonin reuptake inhibitor, so it increases serotonin. And we know the FDA approved serotonin reuptake inhibitor to treat COVID-19 because serotonin inhibits the polarization to Th17, M17. So it's an anti-inflammatory drug, those serotonin. So serotonin reuptake inhibitors, Methylene Blue, are FDA approved. The serotonin reuptake inhibitors, those drugs that we know for treatment of COVID-19, and the mechanism is serotonin inhibiting interleukin-6. So Methylene Blue does everything. I mean, if you see it, it does work on all aspects to manage the acute phase of COVID-19. And if you add the light, you activate, you generate free radicals, you can kill more of those viruses. And it will help to balance the immune system, the metabolic, and the redox when we are facing the post-COVID-19 syndrome and those fatigue and endothelial dysfunction and mitochondrial dysfunction due to the cytokine storm and due to the free radicals that's being generated during the inflammation. That make sense?
Does, yes.
All right. And naltrexone is very good. Dr. William is very advocate for naltrexone low dose because naltrexone inhibits M17 and Th17 polarization. So it's a treatment, naltrexone low dose. And they work the same like peptides, naltrexone. They work on toll-like receptors on the macrophages, and they inhibit the polarization of the macrophages into Th17. So I will add low dose naltrexone in the equation. I will add polyphenols. I will add antihistamine. I will add Methylene Blue. Ivermectin I will use during acute phase, but for post-COVID-19, I would like to use just Methylene Blue and polyphenols and peptides, thymosin alpha. I will not use thymosin beta. The reason I need to check the immune balance or the immune, we call them the lymphocyte mapping and cytokine profile and see where the immune is. And if it's polarizing into Th2 and Th17, you really don't want to use thymosin beta here. You want to use thymosin alpha to restore the balance and the polarization to Th1 and cytotoxic T cells. We know that COVID virus and post-COVID, because of this circuit and vicious circle of activation of Th17 and interleukin-6 and this inflammation and then mitochondrial dysfunction, and also this inhibition of the nicotine receptors and all that, you will end up with a vicious circle of Th17 inflammation. And what will happen is that you're draining your immune system to build or to have neutrophilia or draining the immune system into Th17 inflammation. And what would happen, you would have less of immune surveillance of cancer through Th1 and cytotoxic T cells. So this will put the patient at risk of having cancer. Or if patient is in remission, then they would have recurrent cancer because they don't have much of Th1 and cytotoxic T cells to kill those cancers, because the immune system is being drained and polarized due to this COVID-19, due to interference of adrenaline angiotensin system into Th2 and Th17. So that's why the number of cancer cases will increase after this post-COVID-19 syndrome. And we saw that, and there's articles and publications, in addition to real clinical cases, that Dr. Patel herself have a case with breast cancer due to post-COVID, either post-COVID virus or post-COVID vaccine. I don't see much of difference between them because both of them are expressing this pathogenic spike protein. Any other question? Are we good?
We are good.
So we can do more sessions is better, because I think I did too much today, right? And a lot of
Okay.
and I don't want to, you know, drain your people from the first lecture.
Oh, they can take it.
You want me to continue, or you want me to go ahead and do next session?
Both.
Oh, let's make a vote. Do you want to continue, or you want to have divide into sessions? Sessions or whatever, just text and let us know, and then we count them. I don't see the number.
I greatly appreciate what's being taught, but I think it's very, very worthy of a next session.
Yeah, I think it's better. I don't want to put
Okay.
too much, because it's very stressful, this information, and it requires time for processing and digesting. And I think when you come next time, the next session, we can go through those slides more easy, because we have some basic. Okay, great, beautiful. Okay.
Okay. All right, Dr. Halasa, I'll get with you to get the next session. I just want to say this on the web, not the conference website. If you were at the conference and you have the password, Dr. McCullough's talk that was 2 hours long, whether you like him or not, is on there. And I'm going to put it on the free side too, probably in the next day or two, because I think it's important to listen to it. You know, even if you're 100% against what he has to say, he makes some very good points, especially about vaccine development and safety, and his take on how things deviated in the case of the these vaccines here. So if you were at the conference, if you don't have the password to get to the slideshow, let me know. I think tonight I'll probably just send it out to everybody again. And I'm going to put it on our webinar site shortly, so that anybody can listen to it for free. It's almost 2 hours long, and those of you who were there know that he's quite the dynamic speaker. So with that, any questions, problems, comments? Next week, Dr. Patel will be speaking for us again, and I do need speakers. I have endless Tuesdays to fill up, as I keep saying. So if anybody, and this is a good forum if you're not used to speaking and you want to start becoming a speaker, you want to start speaking. We always need content. You don't have to be polished. Nobody's going to bite you here. Well, maybe Dr. Burgess, but he's mostly harmless anyway. And so, comments, questions, any other questions for Dr. Halasa? Always thought-provoking. And again, if you look in the chat, Dr. Halasa, you'll see an arm's length full of accommodations for you here, as usual.
Well, thank you very much.
Okay. And so Dr.
Halasa, thank you very much.
Yeah,
thank you for listening and attending and be part of this. And we need to hear from you. And I need to hear from everybody. I mean, we have Dr. Patel is active, Dr. Yusuf James. So I'd like to hear from all of you guys, and so we can learn from each other. I mean, Dr. Yusuf, for example, he came up with a lot of new ideas. Dr. Klatz, for example, he said yesterday, hey, we need to really screen those people with the spike protein, and we need to find a test to see how we can measure it in the blood or in the feces. And I told them they're already doing it. They have ELISA tests or chromatographic tests where they can detect the spike protein in the sewage. So in order to know if there's a spike in the infection, they take the sewage of that community and they can detect the spike protein in grease. And if they find the spike protein fragments or whatever they are measuring, then that means that the COVID is increasing. And because of that, then there is tests where you can measure those spike protein in the feces, and you can use it as part of your diagnosis. Then you will know that this fatigue and this sexual dysfunction and this autoimmune or dementia, it could be part of this patient still expressing the spike protein, and those vaccines RNA are not completely biodegradable for some patients. And the virus, they say, you know, it's an RNA virus and it terminates once it's being recovered. It could be those RNA reversed to DNA. We don't know. There's some articles of publication that says that. But shedding more than 3 months or 6 months of the spike protein indicate that there is some way of harboring those RNA, whether reversing to DNA or they are stable and they don't degrade and they continue to express. And I don't see that in all the patients, but some patients. And then we can really measure the spike protein and knowing that this is part of the pathology, this is what you're dealing, and we need to detox those spike proteins. And the best way of detoxing them is using methylene blue, because methylene blue combines with it. I mean, you can use ivermectin, but I prefer methylene blue for many reasons, because it has many other aspects of restoring the endothelial function, mitochondrial function, autonomic nervous system. It also increases acetylcholine, so it's good for dementia. So there's many things about it. And it polarizes the immune system, or it balances the immune system. So that's something that Dr. Klatz is emphasizing and adding it to our protocol. Hopefully that Mike Beamer will be looking the lab to have that as a routine protocol for managing post-COVID-19 syndrome.
Okay. Anybody else?
And Dr. McCullough, I think I can invite him to speak again here. He promised he's coming again. I did invite him to speak at Dr. Clearfield, by the way, in his radio show. And he said yes, and they are arranging for him to speak there. But we maybe need to hear from him, you know, especially with all this changes that's happening, and especially with this new restriction of Johnson vaccine. Just recently, I mean, this is like 3 or 4 days, because of the concern of coagulations and the clotting effect that the Johnson only restricted here in the United States, but not overseas, so they can keep selling it.
Okay.
Any other questions? Everybody's happy here?
We're good?
We're good?
Happy
Okay.
and good.
Okay, Dr. Burgess, we have a few things to discuss.
Yep.
Okay. Okay, everybody. Thank you so much. One last question, Dr. Halasa, you still there?
Yes, I'm here.
Okay. Are the increase in skin eruptions hives from Th17?
What is that? Sorry.
Are the increase in skin eruptions hives from Th17?
Well, it could be a combination of Th17 and Th2. So what I would recommend is doing cytokine profile to determine exactly what's going on. But both can cause skin eruptions. The eosinophilia and neutrophilia damage, as we see in rosacea and psoriasis, and we see in eczema. Eczema is due to Th2, while psoriasis and rosacea Th17. So, and it's very important to know, so we can emphasize on which one I need to inhibit. And maybe they're both, but one is more dominant to others. So I can use both antihistamine, and I can use ivermectin, I can use methylene blue, and all those are antibodies that inhibit, or there's drugs that inhibit the expression of interleukin-6 that's FDA approved out there. They're using it for treating psoriasis. So it's good to do lymphocyte mapping and cytokine profile and do the ratio figure out where it is, and then start working on the drugs that inhibit those inflammation specifically, instead of giving steroids, which will inhibit all those three inflammation, especially with Th1, and then put the patient at risk of having cancer and viral infection. So I don't like to use the steroids all the way. Although if I see everything is high, then I will use a dose of steroids just to get into the edge. And then I will make sure that I will focus on the inflammation. That's where the direction of the pathology is. It makes sense?
Yeah.
Okay.
Okay. All righty. Okay, Dr. Halasa, we'll get together and we'll
And
get
you have a
on
big
for-
crowd. They stay up
Yeah, we
to
had-
now, 27.
We had
That's a
40.
big one, right?
We had 40 at one point.
Here we go. You got the record there. Okay, well, invite them to my show too.
I
So
will.
it's gonna be fair. I invite all my folks, by the way, today.
Okay. All right. One last thing. We get a question about what types of devices for phototherapy, and are they quote unquote affordable?
I think they are. Mike Beamer is the one who's in charge on that. He's not, I mean, it's like selling it for $100 or something, 200. A good piece that's put in your mouth for a patient with COVID. I do recommend it. So you reach out to Dr. Mike Beamer, the pharmacist, Amex Pharmacy. I think he has it. And they got this device because it's being published in Bahamas with Dr. Hepburn, and she has studies and data and publications showing the decrease in the viral load using methylene blue photodynamic therapy, and he can share those publications with you.
Okay.
All right.
All right. So, yeah. So if you don't know, it's Amex Pharmacy in Melbourne, Florida.
Right. Just ask for Mike Beamer, and he will or text me or text Dr. William. He gives you the phone number. And having a pharmacist on your side is a good thing, especially, you know, those intelligent pharmacists. And I admire Mike Beamer a lot.
Melbourne, Florida.
Okay.
Right.
All righty. Okay, everybody. Is this a confidential meeting, Dr. Halasa, or is it open to anybody?
Which meeting?
Dr. Burgess, is this confidential, what we need to talk about, or? You're muted.
Kent wants to talk on the phone. I think he's on the road right now.
Okay.
And I'll call you on the phone too.
Okay. All righty. Okay, everybody. Dr. Patel will be with us next week, and we will certainly have Dr. Halasa back shortly. Dr. Zarin and I are planning the last Tuesday of the month, our sort of mini course. And we are going to be speaking on the psychiatric illnesses secondary to traumatic brain injury. That's our topic. Okay, and you know, since I'm going to be one of the speakers, you know, there will be a heavy emphasis on hormonal balance. So we'll look forward to that. Okay, everybody. Good night. And we'll have the video up as soon as we can here. And look, by this time next week, we certainly should have Dr. McCullough's talk up on the free side, on the webinar side also. Okay, so John, you're going to call me later. Is that?
Yeah, I'll call you right now.
Okay. Okay, everybody. So thank you so much, and we'll see you again, same time, same station next week.
Okay, good night, everybody.
Good night.
Thank you.
Thank you.